We conduct our experiment in the laboratories of the Putzberg Foundation with Dr. Federico Grunbaum, a specialist clinical microbiology and parasitology.
In the first interview with Dr. Grunbaum, Sophia and I went with my brother because he was going to perform his experiment on the bacteria there. The doctor explained how working in a microbiology lab, how samples are taken and you can see them. We also spoke of the experiment we wanted to do.
First of all, we take a sample of the urine of the first jet. And we put the number 1 and also the initial letter of the person that is the urine. Later we do it another time, but now with the second jet, and we put the number 2.
We label the vessels to collect the samples.
Collect samples. Samples number 1. Of the 2 individuals, we put them on the oven of half an hour. Samples number 2. Store them in the fridge. How we learned during the investigation, bacteria do not grow at low temperature, and high temperature multiply.
When we wait, we labeled Petri dishes, test tubes, and slides. After 30 minutes, we took samples and we planted. As you see in the video, you have to sterilize the tool and follow the instructions who had given us the Dr. Fede. We filled the samples with the test tubes. We filled the test tubes and we took them to a machine that will move very quickly to the samples to precipitate and remain below the sediments. They will be around 2 minutes. Then when it's finished, we put the sediments under the microscope and we start looking it.
Now we only need the sediment for see it in the telescope. They explain us that we have to drop very quickly the urine for take only the sediment. If not, if we do it slowly, the sediment and the urine will mix and we lose the sediment.
The sediment was placed on a slide and we covered it with cover slip. Then they explain us how we place in the microscope and how we look it.
In the first samples, were very difficult to see cells under a microscope. To get a better view, we'll stain the slides in the machine. Dr. Fede explains how it works. In the yellow tint glazes, alcohol is purple and pink tint. We introduce the samples into the dyeing machine. The cells were stained in pink or purple, depending on the strength of the wall. With this technique, we can see them and identify them better in the microscope. This is the result of the samples after passing through the machine. Now we can see better the different cells. They tell us that in a sample we can see also squamous cells. We made some pictures of what we see under the microscope. Here we see BGN, that it means gram-negative bacilli.
Here we see CGN, gram-negative coccus. Here we see BGP, gram-positive bacilli. Here we see BGP, gram-positive coccus.
Dr. Grunbaum sent us picture of the Petri dishes after 24 hours. Bacteria can be seen in the sample number 1 of the woman, and the others are clean. We return to the lab of the Putzberg Foundation with Dr. Grunbaum. We take out the samples from the refrigerator number 2 that we had planned in the Petri dishes. The sample number 1 had been at room temperature to continue growing. This is what we found. You can see that our hypothesis is being confirmed in samples number 1, UFC, units of bacteria per milliliter. is much higher than in sample number 2. This is our experiment. We want to thank Dr. Grunbaugh, laboratory staff and Clinical Puigverd Foundation by serving us so well. Thanks. We had learned so much.